Hello everybody and welcome to the IMI webinar on the IMI2 Core 7 topic on the identification of druggable targets modulating misfolded proteins in Alzheimer's and Parkinson's diseases. First of all, I'd like to apologize for the late start today. We had some technical issues which I'm happy to say we have found a workaround for, so we will get going now. I will start off with a little introduction to GoToWebinar so you know how to use it. And then my colleague, Elisabetta Valdano, who is the scientific officer responsible for this topic, will give a brief introduction, including some tips on how to make a good, strong application. After that, Kenneth Seerstrup of Lundbeck will be on hand to give the core topic presentation, and then we'll have questions and answers. So, how to use GoToWebinar. At the top right-hand corner of the screen, you should be able to see the control panel. You can expand and minimize it. You can make it go full screen, and you can attract our attention by raising and lowering your hand. If you want to listen over your computer's mic and speakers, then you just need to select mic and speakers from the option, and if necessary, do a sound check to make sure GoToWebinar is picking up the right mic and speakers. If you want to listen in over the telephone, just select telephone and dial the numbers given. If you want to try additional numbers from other countries or a free phone number, you can find a list under the additional numbers link. If you want to ask a question, you can do that in two ways. The simplest way is to send your question in writing by typing your question into the question panel and clicking on send. Alternatively, you can ask your question over the phone by clicking on the raise hand icon. You can send in questions in writing at any point in the webinar, but we'll take all the questions at the end. So please note that this webinar is being recorded and it will be published on the IMI website or the IMI YouTube channel. We will publish the presentation slides on our website after the webinar, and we'll be circulating a participant list for you to network. So on that note, I will hand over to my colleague Elisabetta, who is going to give you some advice on how to put together your presentation, among other things.
So good afternoon, everybody. And what we will present you today is a webinar that will cover all aspects of the core topic. And so we will give you an overview of the objective of the project, why is a public-private partnership approach needed, so why we are launching this as a part of the IMI initiative, the possible structure of the project that should be generated by this topic, the expected contribution from you, that is the applicants, and the contribution from the other important partners in these initiatives, which are the industry, the FPA participant, and what are the key deliverables, which would be also the really key output necessary for the impact of your proposal. We will not cover today the rules and procedure of IMI. Of course, if there is some specific question, this can be done in the Q&A. But if you want to really know everything about it, you are very welcome to register for the webinar on rule of procedure that will take place on the 20th of January at 3 p.m. And also, if you can manage for that, this will be a presentation and recording will be published on our website. So what could be a few tips for your submission? First of all, please, it is very important that you read very carefully all the relevant material. And of course, the most important is the topic text. And also really have a look to also the introduction and to the final section where there could be some other important relevant information. And when you read it, please take attention to the aspects that are especially the objective, the deliverable, since you will be expected to address all of this. And also when you do it, try to understand also what is the purpose of IMI, that is that you will have to create with the industry consortium a public-private partnership. Then, of course, you should try to understand as good as possible the IMI2 rules, and all the relevant documents are present on the website on the call stage 1 page. But if you have any question, please contact us, and we will answer as fast as we can. But please do not try to contact any of the FPA topic team members, the coordinator or any other members, because this is not allowed. And if this is come out, your application could be disqualified. And when you start writing to your proposal, of course, look carefully at the template and try to provide also all information the reviewer will need for evaluating your proposal. So please have a look to the evaluation form and to the criteria that we use to evaluate. And also remember that you need some time to get a good consortium and to write a proposal. So try to start work as early as possible. When we come to the submission, this will have to be done our online tool, the Sophia tool, and it's very important that when you submit, you remember that you have first to finalize and then submit. If you don't do that, we will never be able to see your proposal, so it will not be able to be sent for evaluation. We have also a list of more tips which are available on our website to the link that is indicated in the page. One important thing, of course, is to allow that your proposal will get to the evaluators is that you respect what are the admissibility and eligibility criteria, because if these are not met, again, your proposal will not go for evaluation. So clearly you have to submit by deadline. You have to be careful and read what is the scope and of the topic, because if your proposal is completely out of scope, then it will not also, of course, go to the evaluators. And you have to use the template that is available for the submission, since if this is not done, then again, your proposal will not be found to be admissible. And also for what concerns the eligibility, please be very careful that for following the rule of Horizon 2020 and IMI2, there need to be at least 3 different legal entities that have to be independent and have to be established in 3 different countries that are of the list of countries that are eligible for funding. And then these are really the minimum to get to the evaluation. And then once you are there, since, I hope you are already all aware, only the first ranked proposal will proceed to the stage 2. If you want to increase then your rate of success, you can really try to think very well, first of all, how you build your consortium, so that not to have redundancy, but of course be careful that you have the necessary expertise to deliver all the objectives. In fact, as is written on the second bullet here, you are expected to address all the objectives of the topic. And once you have addressed all the objectives, you have to consider also that you address it in a way that you can achieve the deliverable, since this is what will be key for the evaluation of the impact of the proposal. So you can have a very excellent proposal for scientific excellences, but if you cannot demonstrate to the evaluator that you can, via this excellence, also really achieve all the deliverable, then the impact will be considered only very limited. For what concerns finding project partners, this is not something that we can help you directly, but we can of course give you some advice. And first of all, it's very important, of course, that you network your contact, and then that of course you have the opportunity now participating with the webinar to be able to network also with a fellow webinar participant. There will be a list of participants that will be distributed afterwards, and people have the opportunity to opt in or out, but those that are in will be able to participate. You can also, of course, ask some question and say something about you when you are now in the webinar. It's also an opportunity, of course. And then for what concern online tool, we have an IMI partner search tool, and which you have the link on the page. And also, of course, what could be also interesting for you is to get in touch with a local IMI contact point, and also ask that they use their network to get in touch with other contact points in the other countries. Then also there is also the Health National Contact Point, and you can also use social media, like in particular the IMI LinkedIn group. And here you see how you can contact IMI.
Thank you, Elisabetta. So yes, today is not your last chance to ask questions about this topic, and if you have any questions, send them to the address there, infodesk@imi.europa.eu. Do not try and get in touch with the FPA partners involved in this topic, because that could raise issues of conflict of interest and unfair treatment. So any questions you have, whether it's about the topic or about our rules and procedures, send them all to the info desk and we'll make sure that you get an answer for that. So on that note, I would like to hand over to Kenneth Thurstrup, who is going to be giving a presentation today. He works for Lundbeck. Kenneth, it's over to you, and because there were some technical issues, just to let you know, Kenneth, we're now on the slide on the need for public-private collaboration.
Okay, welcome to this webinar. I'm Kenneth Thurstrup from Lundbeck, and for the call on this webinar, I'm also representing 4 other companies involved in the topic, namely AbbVie, Novartis, J&J, and Lilly. Manuela Puldure from Novartis, if she has managed to join the webinar, she might also be assist me in answering questions and join the discussion after the presentation. Why launching this call and why the need for public-private collaboration? As you know, there's a high need for new disease-modifying treatments for AD and PD. Treatment used today are in most cases developed from old drugs based on dopaminergic and cholinergic signaling pathways. They are all drugs developed for symptomatic treatments, and drugs which can slow down or stop the progression of the disease are much needed. The latest new AD drug approved goes back all the way to 2003, which was in this case an NMDA antagonist memantine for cognitive improvement. So although a beta, tau, and alpha-synuclein have been known to play a major role in the pathology for years, there is a lack of understanding of how these proteins affect pathology, and this has limited the identification of new targets. Many hypotheses have been brought forward based on more indirect biological links to pathology without a clear understanding of cause and effect. For example, the role of oxidative stress, mitochondrial dysfunction, or neuroinflammation are areas where there is and has been many efforts going on to show they are important for disease progression in AD and PD. But clinical testing of molecules targeting these biologies have not been successful so far. Of course, it's fair to say that there are many explanations for clinical failures in this area, like lack of good disease progression biomarkers and poor clinical designs. Nevertheless, there has been a move during most recent years toward targeting the misfolded proteins directly with antibodies against a beta, tau, and alpha-synuclein, where there currently is the highest hope for breakthrough medicine against these diseases. Recently, the hypothesis has been substantiated that these proteins display prion-like properties, and this has enabled identification of new targets related to AD and PD. In the prion-like hypothesis, tau and alpha-synuclein spread from cell to cell and act as seeding templates to further accelerate misfolding of these proteins. There are several examples of this, and I will not go into details, but just to mention, for example, in one of the first studies supporting this hypothesis, it was seen in patients from post-mortem examination that Lewy body pathology may spread from host to grafted healthy embryonic neurons in PD patients. Also, spreading of tau and alpha-synuclein has been observed in mice model based on experiments with injection of pathological human material. So as the prion-like hypothesis has matured more and more in the scientific community, we believe that this has opened up for new opportunities to identify new important targets on which future medicine may be developed. Due to the complexity of this area and fast progression research, a successful project will demand a strong understanding of fundamental biological processes and access to disease models and tools which are only available through broad public-private collaboration. So if we move to slide 3. So what are the objectives of the current study? As the title describes it, it is to identify druggable targets modulating misfolded proteins. Before I move on, I would like to point out that the term druggable is a term commonly used in the pharmaceutical industry, and it is more or less a description of a biological target for which a drug can be found to bind to with high affinity and also affect the function of the target with benefits for the patient. Very often these targets will therefore be a receptor or an enzyme where a specific small molecule inhibitor can be identified. But of course it can be any target where some kind of effector can be identified. Thus you can argue that there may exist very important proteins out there which are central players in a disease, but if the function cannot be modulated by a small molecule or biologics, they are not considered druggable. It is therefore also important that the applicants think about what kind of targets we end up with and how we will eventually evaluate their druggability. Although we have defined the call text as open as possible with respect to conducting genomic screens, there could also be an advantage in defining the genetic screens against special targets or classes of proteins. But it is up to the applicants to think about this. So going back to the objectives of the current study, more or less there are 3 overall parts in the process. These parts are also reflected in the work packages, which I will elaborate on later slides. So in the first part, the objective is to set up in vitro and in vivo models for spreading and seeding of tau and alpha-synuclein. This is defined relatively broadly, and the readouts for how the cell is handling these proteins can be different mechanistic of phenotypic readout relating to, for example, exocytosis, endocytosis, and mechanisms for affecting protein turnover like autophagy or proteasomal degradation. So we're quite open in the call for which models should be included in these studies. Of course, there may be some limitation and some pragmatic consideration of setting up the models. Some models may be more difficult to set up in vitro compared to in vitro, especially spreading of misfolded proteins from one cell to another may be more difficult to modulate in vitro. In the second part, we will use in vitro assays to conduct genetic or small molecule screens to identify targets and mechanisms involved in these processes. Based on the setup of these assays, the consortium will select the most optimal assays for the screening purposes. Of course, some of these assays may already be available in the beginning of the project, and it is up to the consortium members to decide when the assays are ready to be used in screening approaches and if some assays are ready from the beginning. The third part of the project, based on key mechanisms identified in screens, targets will be validated and selected in vitro and in vivo assays. Here there's of course the limitations of how many targets can be progressed, and for in vivo studies we might select a few targets to knock down or out or overexpress for further studies, or be modulated by tool compounds if they are available. The druggability of the targets will be investigated more broadly by characterizing implications of modulating the targets. As I just briefly touched upon earlier in the presentation, this is about substantiating that the target may be modulated by a small molecule or biologic in a meaningful way, which may later on result in a clinical beneficial effect. There are different aspects of how the cell responds to modulating the target, which can be studied here. For example, how specific are the effects we can get on misfolded proteins, or are other proteins also affected? Are there any compensatory mechanism regulating in the cell which may lower the response of modulating the targets? Are the targets regulating fundamental cellular processes which may be too problematic to deal with? This is just to mention some of the consideration in relation to the druggability and implications of modulating the target. So if we move to slide 4. The consortium will be working in pre-competitive landscape, and tools and results will be made available to the public. It means that the project should be defined and structured and run in a way where there should be as little barrier as possible for sharing the results, not only within the consortium, but also toward the scientific community in general. To achieve the highest impact, collaboration and synergies with other relevant initiatives and consortia will be considered. Of course, we should be aware that there might be other groups and consortia out there working on similar ideas and concepts, which we should also consider so we don't duplicate other efforts. Also, if possible, we should incorporate learnings from other groups. In the call text published, there are some examples of different EU research and innovation programs, which could be checked out and could be beneficial to consider. The consortium consisting of academia and small medium-sized enterprises, and FPIA aims to develop tools that will be freely shared between partners and the scientific community. We are aware that in some cases this may not be so straightforward, especially concerning in vivo models. There may be third-party licenses involved, which can make this problematic. But at least the applicants should think about how to structure the way which minimizes the boundaries on sharing tools and assay. Thus, consortium members should also share existing knowledge to accelerate PD and AD research, and the results should be shared with the public throughout the project period. And finally, regular meetings and workshops will be held to openly discuss results and progress. So if we move to slide 5. So what is the expected impact on the R&D process? There are several outputs from the project which may contribute to the R&D process of developing new therapies against PD and AD. The main output, as it is phrased on the slide, to establish a common preclinical platform of assay based on the hypothesis of turnover, aggregate, and spreading of misfolded pathological proteins. And this will be important in order to reach a consensus of how new treatment principles can be best evaluated and substantiated. So why is this so important, you may ask? So far, the spreading and seeding assays for tau and alpha-synuclein are set up in many different labs in different ways and with different protocols. We also know that it is not always trivial, for example, to isolate and produce the right kind of misfolded protein to make it ending up with displaying prime-like and toxic properties. This is especially if you try to produce misfolded protein from a recombinant form. But even if you start out with pathological tau alpha-synuclein from a human source, it may be challenging to get those to work in a prime-like spreading and seeding behavior. This goes both for the in vitro and in vivo models. It means that within this area, it can be particularly challenging to replicate other results and set a large and time-limiting obstacle for the pharmaceutical industry to validate new targets. So if we within this area could come up with preclinical platform of these assays, it would lower this bar significantly for moving new targets forward, and also for later development of the therapeutic molecules themselves in the future. So therefore, such a platform can form the basis for identification of new druggable targets, which would open up for development of new innovative treatments against PD and AD. So if we move to slide 6. This slide shows an overview of the suggested overall architecture of the project. It should be noticed that this is a suggested architecture. It may turn out that other architectures or innovative project designs could be more beneficial. Thus, if the applicant consultant has another idea for project design, they are much welcome if properly justified. The final architecture of the full proposal will be defined together with the industry consortium. Basically, it is envisioned to consist of 5 work packages, where work package 1 consists of governance and project management, and work package 5 consists of data and knowledge management. The setup of work package 2, 3, and 4 reflect the experimental part of the project, and at overall level it consists of work package 2 with establishing tau and alpha-synuclein assays, work package 3 which consists of the target identification, and work package 4 which is the final phase and consists of target validation and evaluation of druggability. Although in general this architecture may look sequential in nature, some activities may of course run in parallel. In the next 2 slides I will go a little more details about this. So if we move to slide 7. Okay, so let's start with work package 1, which is most about governance and project management. It includes administration of the grant, making sure the project plan is in good shape and being followed, and also coordinating how results are shared internally within the consortium and externally at scientific meetings. For each work package, we are suggesting that a work package leader creates a detailed project plan and engages together with the project team to define the full proposal. The work package leader should also, if considered beneficial, ensure collaboration with other relevant projects and take initiatives to ensure external awareness. Work package 2 is about establishing in vitro and in vivo assays for tau and alpha-synuclein uptake, seeding, and aggregation and aggregate-dependent toxicity. Basically, it is divided into an experimental part on setting up and validating the different assay and model system. And hereafter there's an evaluation phase where the different assays would be compared and further judged for their ability to function as target identification or validation assay in work package 3 or 4. Work package 3 is about identifying target using genetic-based and small molecule-based screening in cellular assays established and selected from work package 2. Available validated assays can be used directly if agreed by the consortium. Just as I said previously, this work package may not necessarily follow the work package 2, but may run in parallel. So in this work package, genetic screens will be performed by modulating specific targets, typically using knockdown approaches by using like SI or shRNA or CRISPR, or supplemented with a small molecule screen. This is done in cellular models with defined mechanistic or phenotypical endpoint relevant for the uptake, seeding, and aggregation. From this genetic screen, new targets, but also possible mechanism affecting uptake, seeding, aggregation of tau and alpha-synuclein may be identified. So we've moved to slide 8. Okay, so let's move on to the last two work packages. Work package 4 is thought to be the last stage in the experimental phase of the project and involves target validation and evaluation of the druggability using in vitro and in vivo assays. The target validation is about modulating selected targets and studying resulting effects on alpha-synuclein and tau and their downstream cellular response and disease phenotype. It also consists of investigating druggability of targets and more broadly characterizing the implication of modulating the target. Finally, work package 5 is about data and knowledge management. Of course, it will be important to establish data format and standards for data collection, data management, and data sharing. Where possible, we should try to use existing technical solutions for data storage and analysis. So if we move to slide 9. So now we move into the area of what the different partners in the project should be willing to contribute. Besides contributing with project management in the work packages, it is also an asset if the applicants have experiments from previous involvement in drug development. These experiments may not come so much from the university group, but could eventually come from the small and medium-sized enterprises if they are joining the application. Obviously, as the project is centered around AD and PD with focus on misfolded proteins, the applicant should possess expertise in these areas and the proteins tau and alpha-synuclein. You may ask why we like to focus on these two proteins and not on other misfolded proteins like A-beta, for example. There are a number of reasons for this. The first is the need to narrow the project so that we do not end up with too many different models to study. Thus, we have chosen the study of two intracellular proteins which currently has the highest interest and priority from the FPA companies, and where the cellular mechanism for handling these prion-like proteins may be shared. Also, you can argue that A-beta, for example, was identified first, and here there are already small molecules progressed in clinical trials like the beta-secretase inhibitors, which can directly affect this protein. In contrast with tau and alpha-synuclein, it seems more challenging to develop small molecules regulating those proteins, and thus there might be a higher need to identify some new small molecule target. Expertise in other neurodegenerative diseases beyond AD and PD with pathologies based on similar misfolded proteins, like in frontotemporal dementia patient where mutated tau play a major role, or multiple system atrophy where alpha-synuclein play an important role in pathology, may also be of relevance, just to mention a couple of other examples. Then important contributions will be derived from expertise in protein clearance, uptake, endocytosis, exocytosis mechanism, and cell responses to misfolded proteins. And contribution are also expected to be from expertise in the relevant models, screening systems, and tools. And it's of course also important that the applicants are willing to make them available for other members of the consortium as needed. If you go to slide 10, I have briefly talked about what we expect the applicants to bring to this project. I'll just briefly touch upon what the FPA project mentors will contribute to the project. The first is project management, where it's expected that the overall coordination of the project is attached to one of the FPA partners. The FPA partners will contribute with joint scientific leadership and contribute with their experience in drug development and AD and PD preclinical and clinical research. The FPA contribution will be around €5 million, covering a 5-year period. These companies have tools which they will be willing to share within the consortium, which could be cellular and transgenic animal models based on alpha-synuclein and tau, but also, for example, stem cells derived from patient fibroblasts. Of course, there might be some limitation on especially the transgenic animals, where, as also said previously, the third-party licenses for use of the animals can impose some limitation. But in spirit, we will try to be as open as possible concerning sharing of models and knowledge. The companies will not share their proprietary small molecule libraries. This is, of course, here they hope later on to be able to develop commercially interesting clinical candidates. However, they might share, for example, tool compound collections of annotated commercially available compounds where the targets are known. These tool compound collections could supplement the genetic-based screening approaches. There might also be contribution from specialized disciplines within the pharmaceutical companies which are not readily available within the academic groups. This could, for example, be advanced bioinformatic analysis of ADME-PK facilities to position a tool compound for in vivo testing. So if we move to slide 11. So what's in it for you? First of all, it should of course be a good opportunity to get funding for your research initiative. You will be involved in an international consortium consisting of academia and companies from the pharmaceutical industry in a project period of 4 years. You will get access to tools from diverse sources which can help progress your research, and you will be in close collaboration with leading FPA companies within the field of PD and AD. Hopefully this should set the framework for quite an interesting and intellectually stimulating environment. It should also provide a good opportunity to grow your network within this area. Then, of course, hopefully the project will be so successful that new targets will be identified, which can lead to development of the future medicine against AD and PD. So we might very well be in a position where we can have a large impact on how this field evolves in the future and in what direction it is going. Also, the premature nature of this project should allow us to hopefully end up with several high-impact publications. So if we move to slide 12. So to sum up, what are the key deliverables of the full project that we anticipate that we will end up with? The way the project is structured, set up, we anticipate that we should end up with a general platform of different models and tools which should allow us to study different aspects of the biology of the misfolded proteins, tau and alpha-synuclein. That is, this platform should end up consisting of robust in vitro assays for tau and alpha-synuclein uptake and seeding aggregation, which is useful for target identification. By robust, we mean that not only should the assays be able to deliver solid data points with low variability, but preferable, we would also like that the assays are robust enough to be readily transferable to other laboratories to enable reproducibility of results if needed. This goes especially for the in vitro assays. In addition, this platform should consist of established model systems for in vitro and in vivo validation of targets modulating seeding and spreading of disease-associated forms of tau and alpha-synuclein proteins. Some of the assays may be overlapping with target identification assays, but the validation assay may especially consist of more complicated assays, for example, based on stem cells or in vivo models, which are not so suitable for initial screenings. This platform of assay is the foundation of the project and is necessary for us in order to deliver on the other key deliverables of the project. Namely, that we end up with completion of genetic-based screens and tools to modulate targeted genes and identify suitable reagents to quantify effect of knockdown or expression. This makes it feasible that findings can be validated in the accepted clinical models in vitro and in vivo. And finally, that will show that druggable targets can be found which reduce the pathological spreading of tau or alpha-synuclein. So with these words, I will conclude the presentation on the topic and will be open toward questions concerning this. Thank you.
Thank you very much, Kenneth, and well done for doing the presentation in the slightly tricky circumstances after the technical issues. So it's time now for questions and answers. And as I explained earlier, there are two ways for you to do that: in writing by typing your question into the question panel and clicking on send, or if you want to ask your question over the phone, raise your hand by clicking on the raise hand icon. Okay, we start with a couple of questions that have come in. Let me just get these here. So one question is, if I can just open this out a little bit, apologies for this. So the first question is from Kostas Vekrelis. Can non-European labs, for example, from the USA, join the consortium? I think that's more one for you, Elisabetta.
Anybody can join, is welcome to join the consortium. Unfortunately, as a line of principle, laboratories that are not in one of the countries that is in the list of eligible countries for funding, they will participate, but at their own cost. In fact, the fact to become eligible has to be, in case, could be evaluated by the evaluators as necessary, but this is only in a very, very exceptional cases. And also, please be aware that if you have a participant from a not eligible country, this will not count in the magic 3 number that you need in order to be found eligible for that your submission is found eligible. So in case should be an extra participant on top of 3 participants from 3 eligible countries.
Thank you, Elisabetta. I will also add, a question came in from one of the webinar participants before the email, also asking about the question of eligibility. And as Elisabetta said, you do need those 3 people, 3 organizations rather, from 3 countries eligible for funding. But more broadly, you need to make sure you have your consortium has the expertise needed to do the tasks as set out in the core text. The next question comes from Ton Reinders, who asks, the project is running for 4 years, but the FP contribution is for 5 years. Can you explain?
Hello, it's me again. I think that... Oh, Kenneth, you want to say, but I think it's
just go on.
Okay, so I think that clearly this is a misunderstanding, that there must be some... We haven't identified anything in the text that said that is the FTA contribution in 5 years. I mean, it's clear that the FTA contribution will be really people that will be working in the project for the maximum duration that is planned to be 4 years, and therefore the contribution will occur during the duration of the project. It is 4 years. It's very important for the applicant to understand that the contribution is not cash, is really in-kind contribution. So people, models that the industry has, and will make it available to the consortium to work together on the project during the duration of the project, so 4 years.
Thanks, Elisabetta. We have a question from Behrouz Youssefi, and this is more for you, I think, Kenneth. Do you see an impact of PET imaging and experimental nuclear medicine contribution in the project, and if so, in which work package?
So PET imaging, it could be in some of the, of course, if you're developing the in vivo model, so that would probably most be in the validation or in work package 2 and 3, where we are establishing in vivo models to visualize the spreading or aggregation of these proteins, perhaps, or the effect of those proteins in vivo. So I could imagine that it could be incorporating it in the project in work package 2 and 3, perhaps.
Okay, we have a question from Charlotte Batley. Do the traditional foreground and background IP provisions apply? This tends to be important for SMEs who are part valued by IP. I'm going to pass this to Elisabetta, but generally, in terms of our intellectual property policy, what I would say is, for the most part, our policy is very close to that of Horizon 2020. There are some small changes, which I'm not going to attempt to explain because I am not a lawyer. But if you are at all interested in the IP issues, which you should be, obviously, I would really suggest you come along to our webinar on the 20th of January, which will really go into that in detail and where our lawyers will be on hand. Our IP policy is also available on our website, and I would also point out that each project's IP policy is agreed before the project starts, so you don't get any nasty surprises. And actually, we found that SMEs do very well from our projects with the protection of the IP policy. I don't know if Elisabetta wants to add anything. No. Okay, so have a look at our website, come to our webinar next Tuesday or next Wednesday rather, and as well, if you have any specific questions on IP, you can send them to us via the info desk email address. Harold Schmidt asks, do you also consider targets that may lead to protein modification and degradation, such as sources of oxidative stress and biomarkers for this?
So you mean whether is downstream from the misfolded protein in the cells, how are they affecting other biologies? The way that we are in thinking in the project is that we should primarily focus on mechanisms which can directly affect the misfolded proteins and not so much downstream effects, so that we are moving away from what has been looked at previously.
Harold has just sent another little message that says no upstream processes that lead to misfolding.
Yes, that could also be interesting to look at in this respect. Clearly, like for instance, chaperone proteins. Could also play a major role here and be interesting to look at. So definitely yes, upstream from the misfolded proteins. That is also something which could be of interest. Sure.
Thank you. We have two questions now, which I think we'll take together because they're related and they're on the call procedures. One is from Marco Feligioni, and he asks, if we propose a project from 3 institutes and this is selected, will be inserted in the Precompetitive consortium, and how will this work? And Carlos Simoes asks, can you clarify the process of enrollment participation of FPA members? Is this only done post-evaluation, etc.? So basically, we have here a 2-stage call for proposals. Before this first stage started, a group of FPA companies, led by Lundbeck, I presume, got together and created this topic text, which was approved by the member states through our states representatives group, the scientific committee, and the IMI governing board. And now that consortium is taking a step back, and it's now your turn as applicants to put together a consortium, really with academics, with SMEs, with patient groups, regulators, other groups that you think might be relevant, but not with any FPA partners. You will submit those short proposals. They will be evaluated by independent experts and ranked. And the number 1 proposal, the people who put that together, that consortium, will then work together with Kenneth and his colleagues from the FPA companies to put together a full proposal. That proposal will then subsequently be evaluated as well, again by independent experts. Obviously, at the second stage, there is no ranking because there is only 1 proposal. However, it is still a serious evaluation, and it is effectively a go, no-go decision. So it's a 2-stage call process. You do not need any FPA companies in your consortium right now. In fact, as we said earlier, you should not contact FPA at this stage because it could lead to a perception of unfairness. So for now, you really need to network with your fellow universities, with SMEs, with patient groups, put together the consortium. Kenneth showed the slides showing what we are looking for in the applicant consortium and what skills you need. So build your consortium on the basis of that. In response to Marco's point of saying if we propose a project from 3 institutes, again, the 3 institutes from 3 member states or associated countries is the legal minimum for your proposal to be eligible. In practice, you may well need more than 3, so don't restrict yourself to 3. Equally, you want to avoid duplication, but 3 is very much the legal minimum. If you don't have 3, we won't even evaluate your proposal. If you have 3 and it's in scope and meets other basic criteria, we'll evaluate it, but to be successful, you need enough partners to be able to do the job well. Elisabetta, do you have anything to add? No.
No, I think I would like to stress the fact that so now you will submit a short proposal which will lead to the selection of the consortium, the winning consortium, the first ranked consortium, with the best possible proposal that is addressing all the objectives in the topic. So please read it very carefully. And then this consortium will be joined by all the members from the FPA company that are listed in the topic text in a sort of in the complete consortium. So it's important that also when you write your proposal, you think about this, considering what Kenneth has already explained will be the contribution and the possible roles that the FPA industry people will be doing. And please, if you have more questions for how this, which work package, which function, this is the moment to ask Kenneth and his colleagues that are on the line.
Thanks, Elisabetta. We have a question again on the topic, and it's from Jocelyn Karadec. If we have already identified inhibitors for a kinase modulating tau phosphorylation, would the development of those inhibitors fit with IMI expectations regarding this call?
So if you means if they can use those inhibitors as part of the project, I mean that will probably depend on what are the IP concerning those inhibitors, whether we should regard them as tool compounds or how we should they use them. I mean in general we would be interested in that, but of course there could be some limitation with some IP concerning it. But in general, yes, I think so.
Okay. We don't have any questions for the moment, so I will just remind you that the call text and all the information you need to apply can be found on the IMI website. I can't emphasize enough how important it is for you to read this. The evaluators, they are independent experts and they are going to judge your proposals against the call text, which is online, and they will use the evaluation criteria, which are also online. So do have a look at them so that you can, because it will give you a lot of clues as to how you should design your proposal. And also I would advise you as well, if you do want to submit an application, then do get in there early. Start working on it early. It does take time to upload all the material you need into our application tool, so don't leave it too late, because anything submitted after the deadline is not going to be evaluated. We talk a lot about the detailed things here, but the basic things like making sure you respect the deadline, the right format, the eligibility criteria, don't forget those either. They're very important. I don't see any more questions, so I think we can close this webinar. Elisabetta, would you like to add anything? No, so I will say thank you very much for Kenneth. Thank you very much also to all of you, and apologies once again for the delay we have at the start. I spoke too soon. We have another question from Rainer Riedel. Do you already have tool compounds available, or would this be something where chemistry could contribute?
I think we haven't really discussed it among the FPA partners yet, what kind of tool compound we might have available. But as I also said, of course, we cannot use our proprietary libraries, but we have in the past used some public accessible tool libraries in screens. So I would expect that we have some of those libraries, and some of the other FPA partners may also have that. So that is something that I would expect, that we might have some smaller libraries which could be relevant for in this project.
Great. Okay. Thank you very much. So thank you once again to all of you. We will be sending around the participant list afterwards, and as I said earlier, the presentation and the recording of this will also be available online for you later today or tomorrow. So thank you very much and good luck with your applications.